Hi, my name is Barbara Fischer. I'm an evolutionary biologist at the University of Vienna. And my talk today is entitled Patterns of Sex Differences in the Pelvis Did Not Evolve de Novo in Modern Humans. This is an image here of a human female and a human male pelvis, and you're probably all familiar with the prominent features of sex differences in the pelvis. The subpubic angle, for example, is much wider in females than it is in males. Also, the dimensions of the pelvic canal, which is the birth canal in females, are wider in females than they are in males. And the sacrum leaves more space in the birth canal in females than in males. It protrudes deeper into the birth canal in males. Now, we think that evolution produced these patterns in order for birth to be possible, because humans need to be able to give birth to really big neonates with large heads. And if females didn't have these enlarged dimensions in the birth canal, giving birth would hardly be possible. But we know very little about when these patterns actually emerged. We know little about sexual dimorphism in early hominins. Here I'm showing you two references about the debate on Australopithecus africanus, and these researchers discussed and debated whether Lucy was actually female. Now, this debate stems from the 1990s, and a couple of additional Australopithecus individuals as well as species have been recovered since. But nevertheless, if there is a scarcity of material and you have nothing to compare your sacrum and hemipelvis with, then it's of course hard to say how a female individual looks like. This is why we came up with the idea of conducting a comparative study between humans and chimpanzees. We thought that this is particularly interesting because chimpanzees, despite being our closest relatives, have much easier births than humans because their neonates are relatively smaller compared to the maternal birth canal than humans. So there's more space in the chimpanzee birth canal. And we collected a lot of 3D data. We collected 3D landmarks. Each of these red points here denotes the location of a landmark on 99 human pelvises and 34 chimpanzee pelvises, roughly half female and half male. And then we conducted a geometric morphometric analysis based on these data. Here I'm showing you the sex differences in this sample. This is already based on the shape data. And I'm showing you the human shapes, the human pelvis shapes in the first row and the chimpanzee shapes in the bottom row. And next to the female and male average shapes, I'm showing you these ultra female and ultra male shapes, which are extrapolations, and they derive from taking the vector of sexual dimorphism, so the sex differences vector that points from the average female to the average male shape in shape space, and then just adding this vector on top of the average male shape that produces the ultra male shape, and the same in the opposite direction to produce the ultra female shape. And we did the same thing for the chimpanzee sample independently. And if you study those two rows now, you may notice that some of those prominent features of sex differences, namely, for example, the difference in the subpubic angle, is also present in chimpanzees. The subpubic angle is much wider here in chimpanzees. You see it better in the extrapolated version. In females, it's larger in females than in males. Also, the sacrum protrudes deeper into the birth canal in males than it does in females. There's much more space here in the pelvic canal in females than in males, just as in humans. Also, the dimensions of the pelvic inlet, the relative dimensions are larger in females than in males. So there's this striking similarity here between the chimpanzee patterns of sex differences and the human patterns of sex differences, which is remarkable because birth is so much more difficult in humans than in chimpanzees. What we did then was we conducted a principal component analysis of all of this shape data together, a joint analysis of the chimpanzee and human shape data, and here I'm showing you the results of this analysis. In the first panel, I'm showing you PC2 against PC1, and in the second panel, principal component 3 against principal component 2. And in the first panel here, you can see the two data clouds clearly separate, and this here is the human data, and this is the chimp data cloud. And each little point here corresponds to an individual, and the blue points are males, and the red points are females. And those big points here in the middle, those are just the average shapes. So this is the average male pelvis shape, and this is the average female pelvis shape. And what you can see immediately here in the panel on the left is that PC1 separates the species. So the chimpanzees land over here and the humans land over here. But what you can also see is that PC2 seems to separate the sexes, and it seems to do so in both species. This is why in the second panel here I am showing you PC2 against PC3, so we've basically removed the species differences. And what you can see here is that the sex means of the chimpanzees, they fall right in between the sex means in humans. So here we have the human male pelvis shape, the chimpanzee male pelvis shape, the chimpanzee female pelvis shape, and the human female pelvis shape. And the sex differences in chimpanzees seem to be almost aligned with the sex differences in humans. This is rather remarkable. It's surprising how well the sex differences in chimpanzees and humans correspond. We tried to visualize this in one more figure. Here I'm again showing you the average female and male pelvis shapes in humans together with the extrapolations. But what we then did next was that we took the vector of sex differences in chimpanzees just based on the chimpanzee sample alone. We scaled this vector, so we changed its length so that it actually corresponds to the equivalent vector in humans, and then we just added the chimpanzee vector on top of the human sex means, and this is what's shown in the bottom row. And those two rows are virtually identical, meaning that the shape differences in the chimpanzee pelvis between the sexes are equivalent to the shape differences in humans in terms of their pattern, but not in terms of their magnitude. So the direction of the vector is the same, but the length of the vector differs by a factor of 2. The magnitude of these sex differences is twice as much in humans than it is in chimpanzees. This clearly means that these patterns, since they are identical between chimpanzees and humans, did not evolve de novo in humans. They must have been present in the common ancestor of humans and chimpanzees, and therefore they must have been present in all extinct Homo species, including the Australopithecines. But we believe that the pattern is probably much older and of mammalian or amniote origin. Why do we think that? Well, because there are species that are only distantly related to us that also have a sexually dimorphic pelvis. For example, the Virginia opossum is a marsupial. It gives birth to tiny neonates, but it has a sexually dimorphic pelvis, and some of the features that differ between the sexes are the same that are sexually dimorphic in eutherian mammals. So we interpret these findings in terms of the theory of facilitated variation. So we think that the machinery, both in terms of genetics and development, that underlies these patterns of sex differences in the pelvis probably stayed relatively conserved in primate evolution and mammalian evolution, and maybe even amniote evolution. But that what changed is the magnitude, and is therefore a regulatory switch that determines the magnitude. And this switch probably adapts rapidly. We do suggest that the switch relates to hormone excretion, probably to estrogen and relaxin secretion, and to the reactivity of the corresponding receptors. Now, I want to thank my collaborators who did this work together with me, and I want to also thank the University of Vienna, the FWF, for funding, and you for your attention. Thank you very much.